Hello students, let's continue our next topic, that is tools of recombinant DNA technology. As we are studying about principles and processes of biotechnology. So in our previous class we see that how a recombinant DNA forms. So here we see that what are the tools or the instruments that are used for making a recombinant DNA and what is this technology. So first the tools are restriction enzyme, polymerase enzyme, ligase, vector, host organism. We have to understand these terms, what are they. Restriction enzyme are of two types. One is exonuclease and another is endonuclease, which is come under the nuclease group of enzymes. So one add methyl group to the DNA. In chapter 6 you have already studied that during capping methyl group is added to the 5' end. So here you can see in restriction enzyme one add methyl group to DNA and another, that is another endonuclease, which cut the DNA from specific sequences. So the first endonuclease is HIND2, which is derived from Haemophilus influenza. The first restriction endonuclease is HIND2, which is derived or extracted from Haemophilus influenza. It has a special quality that it cuts the DNA segment in from a long stretch of DNA. It cuts the specific DNA from its specific sequence or from the recognition sequence, and we can also say this as recognition sequence. So nowadays there are 900 restriction enzymes are discovered from 230 strains of bacteria, and all these restriction enzymes have different recognition sequences. Here one question arises, you must think about what is recognition sequences. Remember, recognition sequence means, suppose I am here giving you an example to understand this. So see, like your E. coli, E. coli, this is Escherichia coli. This is a restriction endonuclease which is derived from the bacteria Escherichia coli RY 13. So this has the special recognition sequence is GAATTC, means first it reads the length of the DNA segment and then cuts the specific sequence. That sequence contains GAATTC. You know, these are the base pairs, and in DNA base pairs are complementary to each other. Simply you see here, this is DNA A T G C A T G C A T G C. These are the base pairs and complementary to each other. Adenine bonds with thymine with double bond, and guanine with cytosine with triple bond. So here this is the recognition sequence of one strand of the DNA. So when we put this restriction enzyme to cut the both foreign DNA and vector DNA, it reads both the foreign DNA and vector DNA and cuts from this sequence in both the cases. What is its importance? Why we use the same restriction enzyme for cutting both the vector DNA and the foreign DNA? As we have to join and make a recombinant DNA, so it must cut the same sequence in both the DNAs. So we understand it through diagram. First you see how E. coli named. What is the principle behind naming E. coli? The first letter is come from genus Escherichia. The first letter from genus. The second two letters from species. The third letter is the strain, and the last number is Roman number in which sequence it is derived. Many strains can be derived, many restriction enzymes can be formed from a single bacteria. So here E. coli is named in this way. The first letter represents the genus, second two letter represents species, third letter is the strain, and the last Roman number represents in which sequence that strain is derived from the bacteria. Same for Hen the two as I have told you from Haemophilus influenza. So first letter is Haemophilus means genus. Second two letter is species influenza. D is the strain and 2 is the sequence of obtaining or finding the strain. So here we should understand how action of restriction enzyme E. coli occurs. Let's see through diagram. Let's see through diagram. This is DNA. This is foreign DNA and this is vector DNA. This is foreign DNA and this is vector DNA. So the foreign DNA has the sequence as I told you GAATTC. Here E. coli acts both on foreign DNA and vector DNA and it reads the or it has the recognition sequence GAATTC. So GAATTC. Here GAATTC. Now see. So complementary to C is G, T is A, T is A is T, A is T, G is C. And same for vector DNA also. So here opposite to G or complementary to G is C, A, T, T, A, G. This is foreign DNA and this is vector. When we add E. coli to it, then it coats from the sequence and leaves sticky end. So E. coli act both on foreign DNA and coats it as like this GAATTC. And here this also we should draw a clear picture for the action of restriction enzyme E. coli. Look. This is foreign DNA. This is vector DNA. So here the sequence is GAATTC. And here also the sequence GAATTC. GAATTC. Or we can write it here GAATTC. Okay. Now see. It's complementary. The complementary to G is C, A is T, A is T, T is A, T is A, C is G. Same here for G, it's C, T, T, A. When we add E, CO, R, I to it, then it coats from the same sequence to both the foreign DNA and to vector DNA. And how does it cut? Look, it cut like this. Here it leaves G, C, and leaves sticky end, and other T, A, T, A. You must think where is G gone? So G will come from the vector DNA because they have to join. And here, same here. So here we can write G A T T C. GAATTC and G remains. And this is like your. So look, it leaves a sticky end. It also leaves sticky end. They will further join. How they join? Look, this joins with this. They join with each other and form, see here, look, now they form what? They form G A A T T C C T T A G. If we see the sequence, then here this, and from here this. So here two different DNA, one from foreign DNA and another from vector DNA. By the action of E. coli, they able to join and form recombinant DNA. And for their joining, here I have written you that ligase. The enzyme ligase help in the joining of both the foreign DNA and vector DNA and makes a recombinant DNA.